On the left is really the sort of thing Chris was talking about. This is the amyloid precursor protein. It's sitting in the plasma membrane. It gets taken into the endosomes where it's cut up into little bits, and that ends up as these 140 and 142 amyloids, which then go to the outside. And then the monomers undergo all those elegant things that Chris was talking about to form the fibrils or the oligomers. But it's these that are particularly toxic in the brain. And what I'm interested in is what actually happens in response to these oligomers. One of the things that seems to happen is that these can actually form little channels in the membrane. In addition, they can interact with pre-existing channels such as the NMDA receptors, which are very important channels in the membrane. And in both cases, these are gating calcium. Now, the concentration of calcium in cells is normally about 100 nanomolar. That's the resting level of calcium. And in response to these oligomers, the calcium concentration in the neuron rises up to 300 nanomolar. And I'll discuss the significance of that in a minute. In addition, neurons, like many other cells, have an internal reservoir of calcium. And in Alzheimer's disease, these two key channels, which are located on this internal reservoir, are upregulated. So they're also pouring calcium into the cytoplasm. And it's this dysregulation of calcium is what we think causes the pathology. And it does two things. Initially, it activates memory loss. So there's a long period in Alzheimer's where you start to lose your memory. But there is no neuronal cell death, and this can go on for a number of years. Later, when the calcium starts rising up above 300, it starts activating the mitochondria, and the mitochondria are key organelles with regard to cell death, because if they see too much calcium, they activate apoptosis and the neuron dies. But this is a much later phase in cell death, in the Alzheimer's disease. And interestingly enough, Chris mentioned about Parkinson's with synuclein. In Parkinson's, it's the death of these mitochondria which cause the onset of Parkinson's, because you kill off your dopaminergic neurons. So now just let me end by discussing briefly the significance of this 300 nanomolar. So here's a normal individual. During the day, the neurons have this resting level of 100 nanomolar. You see something unusual. There is a brief spike of calcium, goes up to 1,000 nanomolar, and it comes back again to 100 within a second or so. But during that brief pulse of calcium within the neuron, there's been a formation of a temporary memory in your working memory. And this is probably located in the hippocampus. So certain neurons will have a temporary memory trace. And this working memory is equivalent to your RAM in your computer. It's what we use in our conscious state to go about doing our different activities. We have to remember we've got to go to a certain appointment and so on. Now, when we go to sleep, in the early phase of sleep, there's memory consolidation. There's a discourse between the hippocampus and the cortex, which is the permanent drive, if you like, the hard disk on your computer. And significant memories get uploaded into the permanent memory. In a later phase of sleep, during REM sleep, when you start dreaming, there's a remarkable thing that happens in the brain. All of the neurons are oscillating synchronously, and the calcium goes up to 300 nanomolar. And what happens, it wipes out all your temporary memories in your working memory. So when you wake up in the morning, your working memory is empty. You go about your daily business. Any significant memories are stored up in your hard drive. Now let's see what we think happens in an Alzheimer's patient. In an Alzheimer's condition, you've got constant amyloid production. You have constant dysregulation of calcium. The calcium is now sitting at 300 nanomolar. Now an early onset patient can make memories. They can easily produce this 1,000-fold to actually form a temporary memory. If you ask an early Alzheimer's what's the time? They say it's 5:30, and Greg's got to talk quickly, so I'll get off. You ask them 20 seconds later, what time did you say it was? They'll say 5:30. So they can remember. You ask them 3.5 minutes later, they won't know what you're talking about. Because that temporary memory is degraded by exactly the same mechanisms, we call it long-term depression, are degraded by the same mechanism because it's constantly on. So early onset people can go about doing their business, they've got a working memory, but it's never restored, it's never made permanent, because they can never store anything long enough to get to sleep to restore the memory. So I'm quite excited about this because we now know that there are certain treatments which can actually overcome this 300 nanomolar treatment. I don't have time to discuss it. There are two things. Lithium, an anti-apoptotic agent known as BCL2, is able to improve memory in mouse models of Alzheimer's disease. And the last thing I'm really excited about, I've just come across this, is vitamin D. The lowest instance of Alzheimer's is at the equator. As you move away, it increases. And there's a major anomaly in Arab women. They have the highest incidence of Alzheimer's in any group because they never see the sun. So I've been looking into the literature about what exactly does vitamin D do. Of course, vitamin D is involved in rickets and so on. And what it does is to increase the pumping of calcium across the intestine. And it does that by upregulating pumps. So I'm really wondering if what vitamin D is doing in the brain, and I've now found out recently that the brain is stuffed full of vitamin D receptors. No one's ever figured out why on earth do they have vitamin D receptors. And what I think the vitamin D is doing is, we know it increases the expression of plasma membrane pumps, extrude calcium. It also dramatically increases the expression of calbindin. This is a calcium buffer. And the brain is absolutely stuffed full of calbindin, because that's the way it miniaturizes its activity by encasing the calcium signals in little brief areas. And of course, calbindin being a buffer will help to reduce this calcium. So anyway, that's just a very brief Cook's tour through what I think is the calcium hypothesis, and I think it's a really interesting possibility. It's this dysregulation of calcium, all induced by these oligomers, which is causing the serious pathology. And I just thought you might like to hear from that, because it follows on directly from what Chris was saying.